So I'm honestly excited to present this work. This is a project I've been working on together with a really brilliant postdoctoral researcher, Federico De Valle, and also in collaboration with an experimental group led by Yaniv. So what we believe in neuroscience is that underlying every animal behavior, there's a particular pattern of neuronal activity. And what we mean by this is that if we had access to the relevant neuronal circuits and we knew how to decode them, we could know what the animal is doing or perceiving or feeling at any given moment in time. This is a very reasonable, fundamental assumption of systems neuroscience, and it's proven true again and again. It's the only game in town, really. But there are some experimental findings from the past decade or so, which provide, I think, an interesting twist to the relationship between neuronal activity and behavior. And in fact, I think pose something of a conceptual challenge. So what do I mean by that? Well, let me illustrate that with a few examples. The first would be spatial memory. So I'm showing you here the floor plan of an apartment, but you could think of your own home. So your home is a familiar environment, perhaps the most familiar environment, and you can navigate through your home with ease. In fact, we know there are dedicated neuronal circuits which aid you in this. So for example, in your hippocampus, there are cells which fire selectively in different locations in any given environment, so-called place cells. and these help you find your way. Another example would be sensory motor associations. So here's a very common one. So this is the light that you would see at a crosswalk. Green means go, red means don't cross. This is something we learn early on. You teach this to your children as soon as they can walk, and you use it your entire life, almost anywhere on the globe. So most certainly there are dedicated neuronal circuits encoding this association. And we know in an experimental setup that animals learning similar tasks have cells actually in their parietal cortex, which encode precisely this association. A third example would be odor memory. So you can recognize different odors. And in fact, in response to an odor or some combination of odors, there are populations of neurons in your piriform cortex which respond in a very specific and unique way. And you can, in fact, decode the identity of those odors based on those neuronal patterns. So now consider the passage of time. Consider your home a week hence, or a month from now, or a year out. Of course, you can still navigate with ease. That sensory motor association of the crosswalk, of course, is still there. And you can still, of course, recognize the odor of coffee. So these are extremely stable behaviors. But we could ask, do these stable behaviors imply stable underlying neuronal representations? And in fact, the answer is no. It's not an empirical question, right? I mean, I'm sorry, it is an empirical question. It's not a theoretical question. And so the answer to this question required technologies that allowed us to track the activity of large assemblies of neurons over long periods of time. And that happened about over the last 10 years or so. So let me discuss that. First, spatial memory. So this is data from the lab of Mark Schnitzer. It was really the laboratory that pioneered the development of miniaturized cameras, which could be mounted on the skull of mice, wake-behaving mice, and allowed for the tracking. In this case, this is calcium imaging of large groups of neurons over the course of about a month or so. So on the left, you're looking at the field of view. Each little circle here is the soma of a cell. The colors here don't matter. Don't worry about that. And what you see on the right is the activity of these cells over time. So the way it's organized is in the top row, you see the activity of the cells against, this is calcium imaging, where the x-axis is position along a linear track, and the y-axis is the index of the cell. Now the cells are ordered in the top row according to their preferred location on day 5. And then the activity is imaged again on day 10 using that same ordering. And what you see is that while on day 5, you can see this very nice sequential activity, on day 10, there's only the ghost of that activity, whereas it seems the activity has become jumbled. Or you could say that there's a decrease in correlation of the activity between days 5 and days 10. And this correlation continues to decrease over time. That does not, however, mean that space is not being encoded for. Because in the middle row, if you take the cells now and order them according to their preferred location on day 20, you once again achieve this very nice tiling of space. But if you look on previous sessions or subsequent sessions, you once again see this drop in correlation. And you can do this again on day 35. So there's some change in the neuronal code here. And this is the phenomenon of interest. So this is referred to as neuronal turnover or representational drift. And I'll be using the term representational drift in the rest of the talk. Oh, there we go. Okay, sensory motor associations. So this is another study using calcium imaging in head-fixed mice, virtual reality. So here the mice are running on a T maze. So over the first half of the track leading up to the T, there's a visual cue, which indicates which direction the animal should turn in order to receive a reward, left or right. And in parietal cortex, you can find cells that look very similar to place cells, but their place cell activity, so to speak, is contingent upon the upcoming motor action, whether the animal turns left or right. In any case, you can see on the right, the activity of these cells, again, this is calcium imaging, organized in a similar fashion to the data I showed you in the hippocampus. So here you can see the activity of the cells ordered according to their preferred location on day 1. You can see this nice sequential activity. Using that same ordering, if you look at day 10, you see that this activity has diffused out somehow. There's been a jumbling of the activity, a change in the representation, and so on. But again, if you order the cells according to their preferred location on day 10, you once again recover this nice tiling. And of course, you can do it on day 20 as well. So this is a very similar phenomenology. This representational drift is seen both in hippocampus and parietal cortex. Third example, olfaction, piriform cortex. This now is electrophysiology. It's not calcium imaging. These are spikes that are recorded, but you see a very similar sort of representational drift. So here you're looking at a pattern of activity, a response profile, if you will, of cells in piriform cortex in response to the presentation of an odor. I forget which one exactly. So if you look at day 0, what you see is the cells ordered. Again, y-axis is cell index, and x-axis is now time. The cells are ordered according to their response valence. So red indicates an increase of firing rate compared to baseline. Blue indicates a decrease. If you use that same ordering on subsequent sessions after 8, 16, 24 days, you see a loss of structure. And after about a month, 32 days, the pattern that you see is completely uncorrelated with the initial response profile. That doesn't mean, however, that there is not a robust response to this odor. In fact, on the right, if you order the cells according to their response valence on day 32, you see a very similar response profile to what was initially seen, but it's just carried along by a different population of neurons or in a different way. One can quantify this by training a classifier on day 0 and then testing it on subsequent sessions. And what you find is the classifier reaches chance level after about a month. So there are other studies that show similar things, but these are all very recent studies, again, from the past a decade or so. Right, what is the mechanism underlying this representational drift? That's going to be the bulk of my talk today. I'm going to focus on the hippocampus. So specifically, what might be the network mechanisms which match the statistics of this representational drift that we observe in experiment? But you know, people are also really interested in knowing what the functional role of this might be, because you have these very stable behaviors. Wouldn't it just be easier to have a stable neuronal representation? Then, you know, we're all done, we can go home. What I'm going to argue, and towards the end of the talk, is I think that there are some compelling reasons to believe that this representational drift is what you should observe if the network that you're looking at is involved in ongoing encoding of new information. Okay, before I go into, well, yes, specifically, what am I talking about? Specifically, I'm going to be talking about mechanisms underlying drift in mouse CA3 during exploration of familiar linear tracks. I'm first going to talk briefly about the data. This is from the lab of Yeni Ziv. This is chronic calcium imaging in mice. Then I'm going to talk about a simple statistical model to fit the representational drift. Now, this model is only going to fit statistics related to active cells. So whether or not a cell is active or not in any given session. But remember, many of these cells are place cells. They have spatial information. The statistical model is not going to capture that spatial information. However, it turns out that the statistics one finds in this model is completely consistent with a large-scale spiking network operating in the balanced regime. And so we'll actually be able to leverage that to map a spiking network onto the statistical model and hence to the data. And in the spiking model, we're going to include a spatial extent of the neuron's response. So we'll have actual place cells with place fields, and we'll be able to describe as well the statistics of place cell drift, place cell turnover. This all works quite well, actually, I can tell you now. And the upshot is that the drift that's observed in experiment is completely consistent with non-specific random synaptic turnover. Okay, so this is the data. This is similar to the previous slide I showed you. Once again, you're looking at calcium imaging in mouse. The y-axis is the cell index, x-axis is space. This is a linear track. You can see the cell's activity ordered according to their preferred location in session 1. And then you can see a drop of correlation from session 1 to session 2 using that same ordering, and then a slower decay over time. And again, you'll see the same thing no matter what session you order on, indicating that there's something like a stationary process going on. Okay, there's some other ways we can quantify this data. So over here in the upper right, you can see a histogram of place field shifts. So on any given session, if a cell is a place cell and it has a well-defined place field location, you can quantify that. And then in a subsequent session, if that place field has shifted, you can quantify that shift. And what you find is that the histogram of those shifts is sharply peaked around 0, indicating that the place fields do not move that much. However, if you look at the peak, it does decay monotonically over time, slowly. So there is a slow shift over time. We can also quantify what's called the population vector correlation, which is essentially taking these figures on the right and looking at the correlation between them. It includes not just the activity of the cells, not just the information about whether or not the cells are active, but also their place field profile. And as you can see visually on the left, in the population vector correlation, you can see a sharp drop initially, followed by a slow decay. And in the inset, by the way, you can see that the fraction of active cells is constant over time. Okay, before I go into modeling, I want to say something about anatomy in CA1. So CA1 pyramidal cells receive inputs from two main excitatory pathways. The first is from CA3. Now, CA3 cells themselves have a significant fraction of spatially tuned cells. So CA1 is receiving spatially tuned input from CA3. CA1 also receives input from layer 3 of entorhinal cortex. Now, you may know in entorhinal cortex, there are spatially tuned cells. You may be familiar with so-called grid cells. Now, most of the very well-tuned grid cells are in layer 2, actually, the projected dentate gyrus. And it seems that the cells in layer 3 are less tuned. So in everything I'm going to talk about for the rest of the talk, I'm going to assume that these inputs are simply spatially untuned. Okay, so what's causing this representational drift? Well, an obvious candidate is just plasticity along these two pathways. So before I go into a mathematical model, I'm just going to look at sort of like a cartoon model and do a thought experiment. What would happen to the representation in CA1 if there's plasticity along, say, the CA3? I'm using CA3 as a proxy for spatially tuned inputs, or entorhinal cortex, say, spatially untuned inputs. So let's look first at the plasticity of spatial inputs. Again, this is just a cartoon for now. So here's a CA1 cell. I've frozen the inputs, which are spatially untuned, and I'm allowing for changes in the inputs that are spatially tuned from session to session. And remember, these sessions are separated by days. So the time scale here is really days. Well, the CA1 cell is summing over a large number of spatially tuned inputs. That means if I look at its subthreshold voltage as a function of position, it has peaks and valleys. Now, there, by chance, there's going to be the largest peak at some location. And if the cell is active at all, it's going to have a place field there. Now, by virtue of the plasticity, these peaks and valleys are going to move. which means if I look at another session, I may have a place field which is simply shifted by some amount, and I can quantify that by this parameter delta. That's one cell. So let's consider a network of cells. So what causes representational drift here? Well, I have some number of cells active in session one. Those are the dark gray ones, and some inactive. And they have a preferred location. They have a place field here. Now, on other sessions, I can have a drift in two ways. So first of all, I can have a cell going from active to inactive, but I can also have a cell having a shift in its place field. That's also going to cause a change in my representation. And so if I look at the population vector correlation, that's going to drop due to these two mechanisms. And the histogram of these centroid shifts is going to be rather flat. I expect these place fields to shift quite a bit. What about plasticity of non-spatial inputs? Here I'm freezing the spatial input, and I'm just allowing for changes in the non-spatial input, which is going to cause a change in excitability of the cells. It's going to shift the subthreshold voltage downwards, hyperpolarize it, or depolarize it. Well, in this case, the peaks and valleys in the subthreshold voltage are fixed, right? So as I move it up and down, if I do have a place field, it's going to be about in the same location. Another way of saying that is that these deltas are going to be small. So in a network, representational drift here is caused by cells falling out of or coming back into the code. And again, the population vector correlation is going to drop. And this histogram of centroid shifts is going to be constant over time, plus or minus noise. Let me just remind you what the data looks like. So the histogram of centroid shifts is tightly peaked around 0, sort of reminiscent of this plasticity of non-spatial input scenario. But it does decay monotonically in time, indicating that this isn't the whole story. Furthermore, the population vector correlation has a sharp drop followed by a more shallow decay, which is not reminiscent of either of these two. So it's not one or the other. Perhaps it's both. Yes, it is. Okay, so now we're going to go into the mathematical model. So here's my population of CA1 cells. Now these cells receive two types of input, right? I said CA3 or entorhinal cortex. Well, we can just call them X or Y. So these are two inputs. These are actually going to be random variables in my network. And X1T just means the input X for a cell 1 at a time T. And these inputs are drawn from very simple distributions. These are Gaussian distributions with a given variance. So sigma squared S for the spatial inputs and sigma NS for the non-spatial inputs. And these are Gaussians with zero mean. So each cell draws two random variables. It adds them, sums them, and it compares them to a threshold. If the input is above that threshold, the cell is active. Otherwise, it's inactive. So this model already gives me the fraction of active cells in any given session, but I need a rule to determine how the drift occurs, how these inputs change over time. So this is how it works. From session to session, I simply draw a new random variable, but I allow for there to be a correlation between random variables on subsequent sessions. And that can be rho S for spatial or rho NS for non-spatial, right? This is a simple way of doing that. So my X at a time T plus 1 is just proportional to X at time T. This is the correlation, plus a bit, which is uncorrelated. And this random variable comes from the same distribution with the same variance. That ensures that the whole process is stationary and that the distributions do not change. So the inputs to individual cells change from session to session, the distributions do not. So this model has a total of 4 parameters because you can normalize by one of them. So the ratio of the variances, a normalized threshold, and the 2 correlations. And you can fit the data, the statistics of active cells from the data, which I'm showing you here very well. Well, what are those statistics? Well, you have the fraction of active cells. You have the percentage of cells active on a given number of sessions. So you can see most cells are active on just 1 session, but some are active on up to 8. There's the survival probability, which is the probability that cells which are active initially continue to be active on subsequent sessions. And finally, you have the overlap, which again, this is only active cells. There's no place cell information here. So this is not the population vector correlation. This is just taking a binary code of cells which are active or inactive, zeros and ones, and looking at that overlap. Okay, well, these Gaussian statistics are actually precisely what you would expect from a large-scale spiking network, which I'm going to move to now. So here's my spiking network model. I have CA1 pyramidal cells and inhibitory interneurons, which are modeled as leaky integrating fire neurons. And I have, to model the entorhinal cortical inputs and the CA3 inputs, I'm just going to take Poisson neurons. Sorry, here we go. So this schematic here is showing you how we do the mapping between the statistical model and the network model. So again, you take the data, you fit the statistical model to the data, and that allows you to extract these parameters, right? The variances of these input distributions, the correlations, and the normalized threshold. And then you can map that onto the network model, because in the network, the inputs that the cells receive are just sums over the presynaptic inputs, which are the synaptic weight times the presynaptic firing rates. And you can calculate these statistics. I should say the inhibition here. So in CA1, there's none or negligible recurrent excitation, but there are inhibitory interneurons. So the pyramidal cells excite the inhibitory interneurons, which then inhibit themselves and also the pyramidal cells. And here the inhibition projecting to the pyramidal cells is sparse and random, but it's chosen so that the in degree to the pyramidal cells is constant. So it does not add to the variance, which makes the calculation easier. This is showing that you can match the analytical calculation to the numerically generated distributions. Okay, so that gives us the statistics for a single session, but we need to know how to do this mapping so that we can cause the drift from session to session, and that's also quite easy. In the statistical model, we had this correlation rho, and in the network model, we just convert that rho into a fraction of rewired connections. Okay, so when rho is 1, which means the correlation in the inputs is 1 from one session to the other, you do no rewiring. But when rho is 0, you do a lot of rewiring. You don't necessarily rewire everything, because you're drawing a new random variable from a distribution for your inputs. Perhaps on session 1, it's 50, in session 2, it's 100. In our hands, we would just then rewire what's left over, which would be 50. So that's not all of them. You can do it a different way, it doesn't really matter. So at this point, having shown you this mapping, I'm sure you're not surprised that we can fit the network model to the data. That's the green stars. But now I want to move on to the spatial information that the network has. And I want to talk a little about the state of the network, the balanced state, or the fluctuation-driven activity. So here I'm showing you a raster plot from a simulation of this network. And x-axis here is time, y-axis is neurons. This is just some fraction of the neurons in the network. The orange line is showing the position of the virtual animal, and the black dots here are spikes generated by the cells. So in this network, the CA1 cells integrate a large number of inputs from CA3. The CA3 cells, a certain fraction of them, are place cells. And we model them, again, as I said, as Poisson neurons with a Gaussian place field. So there's no heterogeneity in the CA3 place fields in this model, but nonetheless, in CA1, in the network model, heterogeneity emerges, okay? So what you find here is sort of a large distribution of spatial information in the CA1 cells. I'm showing you an example of 9 of them here. So the top row are the most spatially tuned, the bottom row are the least spatially tuned. And if you look at the entire distribution of tuning in the network, it looks like this, where it's quantified in terms of spatial information. So how does this come about? Well, it comes about through a mechanism which was already studied by David and Carl a decade ago in a different context. So they were looking at a network model to describe the emergence of orientation selectivity in cortex without orientation columns. So salt and pepper cortex, so to speak. And it's the same mechanism here, okay? Although we don't have recurrent excitation, it doesn't matter, it works the same way. So let me just illustrate that briefly. So I'm looking at a cell here in one of the CA1 pyramidal cells, which has the following spatial profile. So it's sharply tuned, and it receives inputs from a large number of cells, as I said. So here's just an example of some of them. The red ones are sample place cells from CA3. And the blue ones are inhibitory interneurons. So these are some of these leaky integrated fire neurons, which do actually have spatial tuning, but less so. Now, as I said, this cell is receiving a large number of inputs. And actually, if I look at the histogram of the number of presynaptic cells as a function of the cell's preferred orientation, you see that it covers the entire space. In fact, to leading order, the inputs are not tuned. That is, the non-tuned part has a large prefactor associated with it, both the excitation and the inhibition. But in this balanced or near balanced state, this large prefactor, these untuned pieces cancel out in the mean, and what you're left with in the end is an order one tuned component, which then leads to the tuned output of the cell. Okay, so I have place cells in my network. What is the mechanism leading to drift in these place cells? I'm having trouble advancing here. So there we go. So the mechanism is the following. So I have these place cells. And if you recall, due to the fit to the statistical model, maybe I didn't say that. So in the fit to the statistical model, the largest amount of plasticity, so the lowest correlation, is in the non-spatial inputs. Okay, that's what leads to that large initial drop in correlation from one session to the next. The plasticity in the spatial inputs is actually much less. That is, the correlation is much higher. So the largest effect is that you have some place field here, and the plasticity from one session to the next is changing the excitability of the cell. Okay, it's hyperpolarizing it or depolarizing it, and therefore you have a drop in the activity, but the location of the place field is essentially unmoved. Okay, and then you can see here's session 6. The activity just by chance goes up again, and the place field is recovered at the same location. Not exactly the same location, because there is plasticity in the spatial inputs. It's just less, so it takes longer for it to take an effect. And so now this is the distribution of place field shifts from the network model, which you can see is the same as in the what's observed experimentally. You have a sharp peak around 0. But the peak actually decreases monotonically over time, slowly. Okay, so now you can look at the activity in the network as a function of sessions, and you recover something which looks very, very much like the experiment. So again, this is from the network simulations now, not the experiment. So you order the cells according to their preferred location on session 1. Using that same ordering, you look at the activity on subsequent sessions, you see a sharp drop from session 1 to session 2, and then a slow decay. And of course, it's a stationary process. And you're able to fit that population vector correlation from the data. Okay. Well, so this network mechanism actually makes a strong prediction, which we can test in the data, which is that the stability of the cells is correlated with their firing rate. It's actually a very simple mechanism, and this is how it works. So imagine you take a cell from the network, which starts out with a high baseline firing rate. So that's shown here. So this is from the simulations. So this is a cell's firing rate profile as a function of space on session 1. And the dashed line is the mean rate. Okay, the dotted line is the threshold rate that we take for actually determining if the cell should be considered active or not. So you can see that its mean activity is well above that threshold. So if the mean activity is well above threshold, then the changes in excitability of the cell from day to day due to plasticity are not too likely to drive it below that threshold and make it inactive. So it's going to be considered as a stable cell. You'll see that it's actually in the neuronal code over a large number of sessions. On the other hand, if you look at a cell which already has a low mean firing rate, which is close to threshold, then even small changes in its input can drop it below that threshold. And you'll consider this cell to be an unstable cell. So you can calculate the probability that a cell is active conditioned on its activity on a previous session. Okay, and this red curve is actually an analytical curve from the statistical model, assuming an exponential transfer function, which turns out fits very well the network Even though it's not identical to leaky integrated fire transfer function in a limited regime, it actually does a good job, okay? And the green stars are from the network simulations. Well, that's just what you find in the experimental data. Now, we don't have access to the cells that are not detected as active, but the ones which are detected as active show this correlation. That is, the probability of them being active on a session T depends on their activity in a session T-1 in this way. All right. And this is just another way of looking at the same data. If you look at the mean activity compared to threshold, you'll see that the higher that activity is, the more sessions that cell is active on. Right? This is, again, this is the network simulations, and this is the data. Okay, so I've shown you that representational drift in CA1 is consistent with a random stationary rewiring of inputs, and that there are these Gaussian input statistics. And this means really two things. First, that drift is consistent with just random, undirected, non-specific synaptic turnover. This sort of massive turnover is actually observed in hippocampus and other cortical areas. And secondly, that differences in the stability of cells is entirely consistent with just a smooth distribution of inputs, or it could be excitability of the cells. So to describe this phenomenology, at least, there's no need for specialized subtypes of cells. Okay, so now I come to the perhaps more interesting part of the talk. Why should there be drift at all, right? And so what I want to argue is that drift is something you can't avoid. It's something you're going to have if you're looking at a system which is continually learning. And this is not a new idea by any means, but it was recently brought up again in this context, in the context of representational drift. This is the paper on olfaction. And the authors of that study say that what they find is consistent with a model in which piriform cortex functions as a fast learning system that continually overwrites itself. And this is precisely the role that's attributed to hippocampus in the memory and memory consolidation literature. So what's meant by this exactly? Let me describe that if I can. So consider a network, two networks in this case, a sensory network and a memory network, or two layers, if you will. And I want to store a pattern in this network. And what I mean by that is that I have some, in this case, these are going to be random patterns. So I have some pattern of activation in my sensory network and some pattern of activation in my memory network. And I'm starting off with some synaptic connectivity here. I'm only going to consider feed-forward connectivity. And by virtue of this pattern of activation, there's going to be some plasticity, some Hebbian plasticity, which is going to associate, again, Hebbian meaning that the connections are strengthened between cells which are coactive. So what consequences does this have? Well, I just stored that pattern. Now, if I want to test that pattern, that is, if I activate that pattern on the sensory side, given this synaptic connectivity, I'm also going to activate the remaining part of the pattern. So I'm going to perform a pattern completion. Well, that's fine for pattern mu, that I'm calling it. What if I want to store another pattern? So I'm starting off with my connectivity, which is storing the association for pattern mu. And now I'm going to activate the network with a new pattern, mu+1, which is uncorrelated with pattern mu. Well, again, that's going to cause Hebbian plasticity. So now the connectivity has changed. But now I want to test pattern mu again. And remember, this is the sensory half of pattern mu. But now it generates this pattern. That's not the same pattern as before. This was the pattern before. So in this extreme example now, there's no correlation between the pattern that I just elicited in the original memory pattern. Okay, more in general, in this type of network, I might want to store many patterns, pattern mu+1, and so on and so forth. And I'm just going to measure time in terms of patterns. And then every once in a while, I'm going to test pattern mu. That is, on the sensory side, I test with the sensory half of pattern mu, and I see what that elicits in the memory half. And what you find in these types of systems, very generally, in fact, any type of memory system like this, which has bounded elements for storing the memory, you're going to find that this memory is going to decay. Okay, so the correlation with the original pattern is just going to decay over time when I do this testing. And you can try and alleviate this decay by reducing the amount of plasticity, let's say. So there's not as much interference from other patterns. But if you do that, then the initial strength of that memory is going to be weaker. So this is known as the stability plasticity dilemma. It's been studied in neuroscience for a long time now, starting with the work of Dani Amit and Stefano Fusi in the 90s. And people have gone to great lengths to study both at the single synapse level, in fact, and as well as the systems level, how to alleviate this decay or reduce it. So that's sort of a primer on the stability plasticity dilemma. How is that related to representational drift in the hippocampus? So let's get serious and actually do a model. So here's my sensory and my memory networks here. Now I'm calling them suggestively entorhinal cortex and CA1, and I'm going to actually implement a model. So I'm going to randomly stimulate some subset of cells here, and then I'm going to potentiate a synapse between these cells with a probability P plus if they're both coactive. If one cell is active and the other is inactive, I'm going to depress the synapse with a probability P minus. These synapses are just zeros and ones for simplicity. And if the two cells are inactive, I'm not going to do anything. Okay, so this is probably the simplest Hebbian rule that you could use, which leads to sort of a non-trivial connectivity pattern. Additional parameters are the sparseness of activity of these patterns, which I'm calling S and F in this case. And remember, I'm in CA1, so I'm also receiving spatially tuned input from CA3. So there's these are all large networks, by the way, in the order of thousands, something like that. So each cell in CA1 is receiving a random fraction of these CA3 inputs. So the connectivity from CA3 to CA1 is sparse and random. But for the time being, I'm not going to consider plasticity here, just in the left-hand side. So the CA3 cells have Gaussian place fields. So the CA1 cells sum over a large number of Gaussian place fields. So they have peaks and valleys in their subthreshold, in their inputs, let's say, as a function of space. But remember that these sensory neurons here are, I'm assuming they're untuned as a function of space. And the CA1 cells are going to be modeled as just threshold linear units. So that is, I sum over the total input of a cell. I compare it to a threshold. If it's below that threshold, the cell's inactive. If it's above that threshold, then the cell's output is just proportional to the input. So that's my model. And now remember that in order to map the previous models, the statistical model, the network model, to the data, really all I needed to do was know what the input distribution was. Well, in this simple Hebbian plasticity model, I can calculate those as well. And in particular, I can calculate the statistics of the inputs, say the number of connections that a cell receives here, known as the degree. So this is showing you that this is the correlation in the degree over time as I learn different patterns. And the gray line is the theory. The black line is simulation. And then I have these funny red dots, which I'm calling selected patterns. What does that mean? Well, I know from the data how large the initial drop in correlation of the degree has to be in order to account for what's seen in the experimental data. It turns out in this Hebbian model, I have a different time scale. The time scale isn't sessions, the time scale is patterns. So it might be that the number of patterns between sessions is not 1, maybe it's a different number. Here it turns out to be 9. So I need to learn 9 patterns in this Hebbian model to observe the correct drop in correlation to fit the data. And when I do that and I look at the activity in CA1 in this very simple Hebbian model, I see this, which is very reminiscent of what's observed in experiment and the network model. And now this is not fit quantitatively to the data, but because we haven't yet introduced the plasticity in CA3, but rest assured, we can fit it. So I'm nearing the end here. I want to say this is a bit odd because here I have this network which is just encoding a bunch of random patterns. But that's not what the mice are doing, right, in the experiment. So the mice, they're running along a familiar track. And when they're not doing that, I guess they're in their home cages or they're being handled or sometimes they're in a bucket. In some of the experiments, they actually explore 2 tracks, one in the morning, one in the afternoon. But it doesn't seem to be what I'm modeling here. But what I would argue is that this storing of patterns is more related to the passage of time than anything else. And so I hope to convince you of this through introspection, which I know is not always the best approach in neuroscience. But if you think of, today's the 2nd of February, Wednesday, try and recall, if you can, what you did yesterday. Maybe you can recall a meal like dinner. I usually remember meals. Maybe you recall a conversation you had, or a plane that you saw flying overhead. We remember snapshots from the recent past for no reason, seemingly. I didn't try to remember what I had for dinner. I just do. But try and remember what you had for dinner or what you did a week ago today. Harder, maybe very hard, or a year ago today, right? February 2nd, 2021, what did you have for lunch? No idea. So I think it's quite clear that our memory system, at least, very promiscuously takes snapshots of our awake experience without us really having to try. Right? These are not memories like the birth of a child or a wedding or some traumatic experience. These are just uninteresting things which are continually encoding. And the reason for this may be that the salience of different experiences is not always immediately evident. So I may eat something today which sits badly with me tomorrow, and it might be beneficial to remember that. It certainly might be beneficial to remember what's been happening to you the past day or so. And so I would argue that something similar may be happening with the mice, even though that they're in this socially impoverished environment. Their nervous systems are still behaving the way they've always behaved. They're remembering snapshots of their experience. And then there's this ongoing synaptic plasticity because of that, which you cannot avoid. Okay, my concluding remarks. So I've shown you that modeling reveals, our modeling at least, points to ongoing and non-specific synaptic turnover as a very likely mechanism for this representational drift in CA1. At least it's entirely consistent. This drift is reminiscent of, it looks a lot like this theoretically predicted overwriting in memory systems, which is so fundamental. But importantly, point 3, I really haven't said anything about this paradox of stable behavior. So, I've talked about how the mechanisms underlying drift, why that might be going on, but you still need to be able to generate stable behaviors. And I haven't explained how that might happen. And I think a likely answer is the interaction of these fast learning systems like hippocampus with slower learning systems in the neocortex, and the interaction between these two. But that, of course, I think would be the topic for a different talk. So with that, I really want to thank Federico. It's been a great pleasure working with him. I've had a great time. I also want to thank David and Carl in particular, because I know some people, during the whole pandemic thing, some people have really enjoyed all the online stuff. I have not. I don't like online conferences, and there's nothing sadder than an online social event at a conference. It's really sad. But there have been a few exceptions, and I think this seminar series is one of them. So I hope that when the pandemic is behind us, this will continue, because I very much enjoyed it. And so with that, thank you all.